Chromosomal theory of inheritance. Till 1900, Mendel's work was not noticed much, and even there were few rejections towards Mendel's work. What were the reasons for that rejection? First thing was communication, as communication was not easy at that time. Second reason for the rejection of Mendel's work was that he stated that genes are the stable units or controlling factors for the expression of traits. But when his contemporaries worked, they found that there is continuous variation, there is continuous inherited variation. So how can the genes be the only controlling factor or discrete unit? Apart from this, the third reason for the rejection of Mendel's work was that he used mathematics. Now the application of mathematics or statistics towards biology was somewhat new and unacceptable. People found that it is not worth, or it is not it's a process to use mathematics in biology. So these three were the reasons why Mendel's work was rejected. But in 1900, three scientists named as De Vries, Correns and Schimak, they again worked and rediscovered what Mendel discovered earlier, and apart from this, there was advancement in microscopy. Thus, they were able to conclude that there are certain structures found which just double and divide before any new cell division. These structures were later found or named as chromosomes. After these three scientists, Walter Sutton and Theodore Boveri, they worked on further this, and they found that behavior of chromosomes is parallel to behavior of genes. These chromosomes, they are paired and as well they separate. They also observed the pairing and separation of these chromosomes, and they concluded that two allele pairs are present on the homologous site of the homologous chromosomes, and chromosomes are doubled and divided just before the cell division. And thus Walter Sutton gave us the chromosomal theory of inheritance, that is, chromosomes are the carrier of genetic material. Chromosomes carry the genetic material, hence they are the basis of inheritance. Linkage and recombination. Now, what Morgan did, he studied Drosophila melanogaster, which is a fruit fly. What he studied is how chromosomes separate and segregate to give inherited characters or to continue the variations. What he did, he took yellow-bodied white-eyed female and crossed it with red-eyed male, and to his surprise, he found that two genes do not segregated independently. Now, what is the reason? He knew that the gene was present on X chromosome. So, he found that in a dihybrid cross, when genes are on same chromosome, the proportion of parental gene is higher than the non-parental combination. So, he came to know that if two genes are closely located, then the parental combination or the chromosome proportion of parental gene combination will be higher than the non-parental one. But these genes may be tightly bound. If these genes are tightly bound, then this linkage will be lesser, even though they might be located on the same chromosome. And if they are loosely bound, they may be located on the same chromosome, but we will observe recombination. So, thus he gave us this term linkage. He gave us this term as he saw physical association of gene on a chromosome. So, he said it is linkage, and on the other end he also gave us another term, named as recombination, that is generation of non-parental combination. Now his student, he further studied and found that this frequency of recombination of two gene of this characters or traits depends upon how the two gene pairs on same chromosomes are located, and he said the two gene pairs on same chromosomes, then he measured the distance, but the frequency of recombination will be equal to two gene pairs on same chromosome and the measure of distance between these two genes on that chromosome, and further the genes were mapped on the chromosome, and these gives us genetic or gene maps. These days these genetics and gene maps are the key towards genome projects, and the latest and the most advanced genome project which is under study is human genome project.